Hello, my name is Priya Sekhri, and today I'll be talking about my work on the modulation of chaperone-mediated autophagy for combating aging disorders. All right, here's a little bit about the person behind the presentation. My name is Priya. I am a rising senior at DeBakey High School for Health Professions in Houston, Texas, and I love spending my free time in the lab. I have been an intern at both the Albert Einstein College of Medicine in New York, as well as the University of Texas's McGovern Medical School here in Houston. I've been a member of many professional organizations. However, my favorite two memberships have been Sigma Xi, the Scientific Honor Society, and HOSA, in which I've competed in the Pathophysiology event for the past 2 years and have been recently named an international medalist. As for extracurricular activities, I love helping my community, truly. I'm a climate change ambassador through my school, and I'm also the executive director and founder of Self-Taught Bio, which is a free educational resource for biology students of any learning level. I also love to write. I'm the author of a best-selling children's book on pollinator preservation and the editor-in-chief of our school newspaper and blog. My dream career is to become a cardiothoracic surgeon in order to help advance global health by treating the number one cause of death globally, which is actually cardiovascular disease. All right, speaking of global health, the world is currently witnessing a demographic milestone. We have more older people than children and more people at an extremely elderly age than ever before. By the year 2050, it is projected that 1 in every 6 people will be over the age of 65. While this reflects undeniably a remarkable advancement in public health, it also presents enormous challenges. Longer lives certainly do not come with extended health spans. They come with a higher burden of chronic degenerative diseases like Alzheimer's, Parkinson's, cancer, etc. These diseases are not only incredibly difficult to treat, but incredibly costly to manage, particularly in nations already strained by limited healthcare infrastructure. But the question remains, what drives this rising burden of disease as we age? The answer begins at the cellular level. One of the key culprits in the precedence of aging disorders is something called cellular senescence, which is a transient response to cellular damage. Under normal circumstances, senescence is a protective mechanism. It prevents damaged cells from proliferating, acting as a safeguard against diseases like cancer. However, when senescent cells persist and accumulate, they become harmful, releasing pro-inflammatory factors called cytokines, which disrupt tissue function, making the aging body more susceptible to disease. Chronic accumulation of senescent cells accelerates aging and contributes to almost every chronic disease that we associate with old age. Fortunately, our bodies are not defenseless. We have mechanisms designed to fight the cellular damage, one of the most important being autophagy. Autophagy quite literally means self-eating, and it is our cell's natural recycling system. It is a fundamental pathway involved in the degradation of cellular components in lysosomes, and it is crucial for maintaining normal physiology. However, as we grow older, our autophagic activity declines, promoting the loss of organ function due to the persistence of senescent cells. Among the types of autophagy, my work focused on a particularly selective and impactful process, chaperone-mediated autophagy, or CMA. CMA is like your cell's quality control department. It is a highly precise mechanism in which the cell selectively binds and degrades soluble cytosolic proteins that possess the KFERQ motif, as you can see on my screen. The protein binds to a chaperone complex in the cytosol that escorts it to the lysosome surface. From here, the protein is brought to the LAMP2A receptor, which unfolds the protein and allows it into the lysosome, where it undergoes complete degradation. Due to the critical role CMA plays in cell-specific functions, failure or decline in CMA is associated with several aging-related disorders. Given CMA's importance, I wanted to answer a fundamental question in my research. How does CMA behave in a controlled model of aging, and can we use this model to control senescent cell accumulation by sustaining CMA activity? It turns out I was not alone in my interest in CMA's role in curbing the detriments of aging. In fact, previous studies have been done to test the chemical induction of CMA activity. Senescence in these models was successfully achieved over a period of 7 days using the chemical stressor palbociclib, and CMA activity was indeed upregulated. However, the results of the chemically induced model of senescence may be confounded by the direct impact of chemical stressors themselves on CMA. In order to mitigate the possibility of interference, I created a novel model of senescence that is genetically controlled instead to determine if CMA is actually upregulated. I also focused on a few specific questions that you can see on your screen. 1. Can we establish the model of genetically induced senescence in cells expressing a CMA reporter? 2. Does CMA activity change in response to genetically induced senescence? And 3. What are the kinetics of CMA changes in genetically induced senescence? In my model, I worked with mouse embryonic fibroblasts, genetically engineering them to delete the TRF2 gene, which protects telomeres, the caps on our chromosomes that shorten as we age. Removing TRF2 leads to rapid telomere damage and triggers senescence. Along with the TRF2 deletion, to visualize CMA activity, I introduced a fluorescent reporter protein called KFERQ dandra, which makes lysosomes glow when they engulf the target proteins, giving them a direct visual readout via immunofluorescent quantification. With the system in place, I was able to closely observe how CMA responded as cells transitioned into aging. In less than 48 hours deleting TRF2, I observed a clear and rapid onset of senescence validated through multiple biological markers, such as an increase in the P21 protein, a decrease in the HMDB1 protein. Additionally, the dendroprotein levels decreased, indicating the upregulation of CMA, as this means that the fluorescent proteins that we introduce are being engulfed and degraded by the lysosomes, which makes them glow. Next, I monitored the kinetics of CMA activity via immunofluorescence. This method involves using the amount of fluorescent puncta observed on each day as an indicator as to how fast CMA was allowing the reporter itself to be engulfed. In terms of results, I observed a gradual increase, as you can see here on the right side of my screen, as to how fast CMA was allowing the reporter to be engulfed. In terms of results, I observed a gradual increase in the amount of fluorescent puncta, which represented lysosomes glowing with CMA-targeted proteins. As you see, they're very pretty. This provided direct visual confirmation that lysosomes were actively engaging with CMA at a much higher rate during senescence. In fact, by day 5, senescent cells were displaying CMA activity at a statistically significant level compared to normal cells. Now, in summary, compared to previously chemically based models, my system demonstrated a threefold faster response time in triggering both aging and CMA activity. Additionally, the level of CMA in this model was much higher than the chemically induced models. This efficiency opens up new doors for studying the relationship between aging and autophagy. But why does this matter for global health? The answer reaches far beyond the lab. Aging is no longer just a biological phenomenon. It is now a defining global health crisis. In wealthier nations, aging populations are ramping up healthcare costs through prolonged treatments for chronic diseases. But it's often worse for our low and middle income counterparts as they're aging too and often much faster, facing both the burden of demographic shifts rapidly towards older population and limited healthcare infrastructure to manage these complex disorders. In many parts of the world, the reality is simple. There will never be enough hospital beds or doctors to meet the demand for late-stage care. That is why preventative cellular level interventions like sustaining or enhancing CMA are not just vital, but essential. I would love to thank my incredible lab at the Albert Einstein College of Medicine's Developmental and Molecular Biology Department. I'm so grateful to Professor Ana Maria Cuervo, who gave me this opportunity, Dr. Susmita Kaushik, Mr. Antonio Diaz, and Ms. Rebecca Sarita. This work was supported by the Einstein Evolution Foundation Partnership on Senescence and Aging. And finally, thank you so much to the GHLC for giving me this opportunity to present, and to my audience for your time and your attention. I hope you enjoyed this presentation as much as I enjoyed giving it. If you have any questions, please feel free to contact me. Thank you once again.